to the macrophage, okay? Not all of them end up with the macrophage looking like this. So we're going to talk about a little cartoon that I drew about what happens with the particle interacting with a macrophage. First, macrophages have proteins on their surface called receptors, which actually can identify a particle. It's amazing. But macrophages will grab onto anything, even including little balls of rubber or whatever. And so they have these receptors, and here comes a particle. And the particle goes and binds to the receptor. Now, the macrophage then goes and responds to that by sending out signals which produce, for instance, the activation of an enzyme called NOX2. Okay? And NOX2 actually generates the superoxide radical. It reduces oxygen, okay, and to superoxide. And that happens actually on the outside surface of the macrophage. Okay? Same place where the particle is. And what happens is the superoxide doesn't last very long. It is immediately converts to hydrogen peroxide. Okay? Gets that second electron. All right, so that's one thing that's happening. Well, what happens next can make things a lot worse. Because remember, a lot of those particles are going to have iron on their surface. Iron is abbreviated Fe, okay, for ferrous, not the Ferris wheel, but the ferrous of Latin, meaning iron. And it goes back and forth between two valence states. For those of you who remember high school chemistry at least, it goes back and forth between iron 3 and iron 2. And it's iron 2 that we really have to worry about. So how does it go with iron 2? Well, superoxide could do that. It could react with it. But the thing one of the most common things that we have in our lung that comes out of our cells, macrophages and other cells, is a vitamin called vitamin C, also called ascorbic acid. It is a great reductant. And we always think of it as something that's really good. And it is good. It prevents scurvy, as we heard earlier today, right? It's a vitamin. It's really essential. And is a great antioxidant. Except if you have metals around, it then helps get things going in the wrong direction. Because what vitamin C does is it reduces the iron from iron 3 to iron 2. And then that iron 2 reacts with the hydrogen peroxide to produce that terrible thing, the hydroxyl radical. That's the really evil molecule, hydroxyl radical, because what that will do is then oxidize lipids, the components that make up most of the membrane of your cells. Okay? That's not good. If enough of that happens, the cell will break open and die. But what happens with small amounts of particles, not real big amounts, really small amounts of particles, is that little bit of lipid peroxidation causes the membrane to signal to make the cell to make what are called inflammatory mediators. Those are the things that will cause the other white cells to come in from the blood, and it's other things that will happen that are not good to happen in the lung. Okay, so secondary organics, you heard about some of them. In the body, interestingly, we have enzymes that will do some weird chemistry to some of these things, like quinones, which we'll hear about somewhat later, also from the Fresno group, I think. But take this quinone that we show here. It can be reduced by a single electron and becomes what's called a quinone-free radical, or a semiquinone. And then that can react with oxygen to produce superoxide. And it goes back to being a quinone. Now, the bad thing about this is that just keeps going on and on and on and on. See, this is the kind of thing that happens in biology, is that reactions start and they just don't stop, okay, necessarily. So this one just continuously makes the superoxide, which could make the hydrogen peroxide. And if you have any metals around, that's why this kind of compounds can be toxic and can initiate either, if a high enough amount, outright death of the cell, or in low amounts, will cause the macrophage, for instance, to produce inflammatory mediators, things that cause inflammation in the lung. Okay? Quinones, some of them actually can also add directly onto proteins, which is not necessarily good if that protein's function is destroyed. And then we have that compound Paraquat, which I showed up there, which has some similarity in chemistry to the quinones. It also goes and constantly causes generation of superoxide. And then another secondary organic, this is, remember, it was mentioned there's thousands of these. Well, here's one class. The molecule in the middle is called acrolein. Some people call it acrolein. Okay? This is one of the kind of compounds that's produced in atmospheric chemistry. All right? Industry produces a lot of this compound. I'm not going to go into all this chemistry here, but one of the things you can see on the top is that it adds to a base called guanine. That's your DNA. That can cause a mutation. So compounds that do that kind of thing, they could cause cancer by causing mutations in DNA. Okay? Along with all the other chemistry that it does as well. All right. So, we're going to talk the last part of this talk, and I'm trying to catch up a little bit for our timing. The last part is going to be a little bit about the research that...
Dr. Foreman?
Yeah.
We're going to give you your full speaking allotment.
Okay.
We may take 5 minutes out of the Q&A. I'll just let you know that.
Okay. So I'll try to... I'll slow down a little bit. All right.
Didn't call for that.
All right. Increases in inflammatory mediators by particles. This is something that I'm working on. Ken Pinkerton here from Davis is also involved in this project, as are some of my colleagues at UC Merced. We are trying to determine the role of particle chemistry in producing the inflammatory mediators. We're also interested in determining the signaling events that go on in a cell. In other words, how does that oxidation of the membrane that I told you about then cause the production of those inflammatory mediators? Now, one of the reasons it's important to know that is that we have individuals in our society who have asthma or other respiratory diseases who are particularly susceptible to particles. There's arguments about whether particles cause or don't cause disease directly, but they certainly exacerbate pre-existing condition. And one of the things that it does that is by producing these cytokines or inflammatory mediators. And so we're going to look at a little bit of that in terms of some data. And I've designed this in a way that it should be understandable even to the non-scientists here. Okay. So we have two mediators that we look at. Okay? One's called TNF-alpha, one's called IL-1 beta. You don't have to know what they do except that it's not good. Okay? And mRNA is just a way that you're looking at on the left side, the left axis, is basically a measure of how much of the genes for these things we're making. So we just take macrophages and we give them small silica particles, okay? That would be from the kind that you would get in the dust. If it's small enough to get down your lung, it can cause respiratory disease, okay? And fibrosis, for instance. And part of that process of starting that is the inflammation. So you can see that these macrophages made, in response, they went fourfold or more. They increased their production of these inflammatory mediators when given the particle. But if we took the particle and we treated it with acid, we can remove the iron. And that tremendously decreases the production of those inflammatory mediators, saying that the iron on the surface is largely responsible. Now, we can't get all of the iron off by that, okay? If we would get all the iron off the particle, we wouldn't have a particle left, because it's part of the structure. This is a comparison. This substance, the thing that produces the brown bars here, that's a substance called endotoxin. If you're anywhere near feces, like near a farm, you're going to get some of this. You're going to inhale some of this. This is the most potent inflammatory mediator we know of.